This lecture will continue into the cardiac chapter. We'll be looking at the conduction system of the heart and also talking about the EKG. Let's review what we've established so far. First, no nerve is required to activate to start the heartbeat. Instead, if you look up here in the sinoatrial node, you will find our specialized nodal cells acting as a pacemaker. These cells spontaneously depolarize about every 0.8 of a second, setting the standard heart rate. Next, these cells here, because they're connected to everything else through those intercalated discs and gap junctions, will spread out this wave of depolarization, starting at the top of our atria, spreading from the top of the heart down. Next, we can look at our conduction system. These are specialized muscle cells that will help take our action potentials, our signals, to where they need to be. The problem is we can't just contract the entire heart at once. Even though these events are very fast, they take under a second, we do need to contract each part of the heart in a specialized order. Let's remind ourselves of the basic cardiac anatomy. These top two chambers are our atria. This is where blood is entering. We're going to contract these two first, and we can contract them together. On the right atria, blood is returning from the vena cava. On the left, blood is returning from the lungs. The two chambers down here are our ventricles. We can also contract those together. The ventricles will eject blood either through the pulmonary vein or up through the aorta from the left side of the heart. The entire conduction system starts in the SA node. The next structure is called the atrioventricular node that lives right between the two atria and the ventricles. The ventricles are also separated by the atrioventricular valve. Once the signal reaches the atrioventricular node, it'll travel down to the apex, or the pointy bit, of the heart. First through our atrioventricular bundle, and then after the split through the right and left bundle branches. Once the signal reaches the apex, it'll spread out through the ventricle tissue by Purkinje fibers, ending up at our papillary muscles. Remember, these muscles, they do not close the atrioventricular valves, but they do hold these tendinous cords in to prevent these valves from flapping back open into the atria. Let's follow the progress of the signal through the cardiac tissue, starting up at the SA node. The nodal cells at the SA node will depolarize about every 0.8 of a second. Our wave of depolarization, our signal, will spread out from the top of the atria down. The atria will start contracting from the top down. This serves to push blood from the atria into the ventricle. This effect is minor. It's only about 20% of the ventricular blood is due to the atria contracting. The signal, after traveling through the atria, will end up at the atrioventricular node. At the juncture between the atria and the ventricles, the heart's fibrous skeleton is very tough and thick. The fibrous skeleton does not conduct action potentials. This forces our action potentials to go through the AV node. The AV node delays the signal, not for long, maybe about 0.2 of a second. This gives the atria time to finish their contraction before we send the signal down to the ventricles. After the signal delay at the AV node, the signal will very quickly pass through the atrioventricular bundle. This is not nervous tissue, but it is specialized cardiac muscle tissue that takes action potentials very fast from one place to the other. They are, in effect, acting as neurons. Going through our interventricular septum, the AV bundle will split. After the split, the signal is taken through the left and right bundle branches, all the way down to the apex of the heart. Once the signal ends up there, specialized fibers called Purkinje fibers take the signal and distribute it around the apex of the ventricle, moving from the apex towards the base of the heart. This serves to contract the ventricles from the apex going up. This is just like squeezing a tube of toothpaste from the bottom up, which is going to be a much more efficient contraction. While this is occurring, our Purkinje fibers take the signal very fast to the papillary muscles. The papillary muscles contract, and this will tighten up our tendinous cords, or the chordae tendineae. This will prevent our AV valves from flapping back into the atria. As with many other things, problems can occur. What happens if your SA node is damaged, either by physical damage or disease? Remember, most of our nodal cells live up in the SA node, and these are what starts or initiates each heartbeat. If the SA node is no longer working, the AV node can take over. Some nodal cells live here. They usually depolarize at a slower rate than the SA node, but if the SA node is not working, the AV node can initialize the heartbeat. The atria won't work as well, but the ventricles will work fine, and the effect on the efficiency of the heart is small. If both nodes, however, are non-functioning, nodal cells scattered around the rest of the tissue can still spontaneously depolarize. If they start the heartbeat, it's going to be uncoordinated and very inefficient. Another problem can occur in what's called fibrillation. This is when the entire heart, or just the atria or the ventricles, are very disorganized and action potentials are flying back and forth randomly. One way to fix this is to completely depolarize the entire structure. And that's what you're doing with those defibrillators. You're sending a shock, an electrical shock, that depolarizes every single cell in the heart. Which cell should depolarize first after that? The hope is that an SA node nodal cell, the pacemaker, will take over and start the heartbeat in its coordinated fashion. However, this only works rarely. How can we assess the electrical activity of the heart? This is done by an electrocardiogram. You'll see both EKG and ECG, and they both mean the same thing. The K comes from the German spelling of cardiogram, but most people actually use this one. One thing to note, this is a composite tracing of all the cardiac activity. This is not a recording of a single action potential. Essentially, several leads are attached, typically at the wrist, ankles, and chest. But those variations can vary, and lots of different locations can be used. So you're collecting a composite tracing of all the action potentials occurring. So don't get confused. The EKG is not the same thing as a single recording of an action potential. This image here shows a typical tracing of your normal EKG. We have several waves and several segments. We will need to figure out what each one means. We can correlate our different waves and segments with events in the total cardiac tissue. For example, let's start at the P wave right here. This activity represents atrial depolarization. So this will be a recording of all the action potentials going on in the atria right after the nodal cells activate the atria. Next, we have what's called a PQ segment. This delay here is going to be the end of the action potential. This will actually be the plateau phase of our typical cardiac action potential. But also don't forget that atrioventricular node delays the signal from the atria to the ventricles. So this section right here will typically be about 200 milliseconds long. Remember, this allows time for the atria to complete their contraction, pushing blood into the ventricles. Once our signal goes down the bundle branches and the Purkinje fibers, we will then start our ventricular contraction. This is represented by our QRS complex. Once again, this is not the same thing as one action potential. That's why it first goes down, and back up again. This just is all of the ventricular cells starting their depolarization phase. Next, we have the ST segment. This is the end of the ventricular cells' action potential. This is the plateau phase of those cells. After the plateau phase, we have our T wave right here. This T wave is representing the ventricular cells' repolarization. So once again, P wave, atrial depolarization. PQ segment, the delay, that atrioventricular node delay. QRS complex is the start of ventricular depolarization. ST segment is going to be the second half of the ventricular cells action potential, that plateau phase. And the T wave right here is the ventricular repolarization. There's one thing missing here. We have an atrial depolarization, but where's the atrial repolarization? It's actually buried in this QRS complex. Remember, the ventricles, there's so much more tissue The depolarization of the ventricles masks the repolarization of the atria. This image shows the EKG overlaid with two cardiac cell action potentials. Between here and here, electrical changes of atria. This is our P wave, atrial depolarization. And then this gap here is this plateau phase of these cells. Here is atrial cell repolarization, but it's buried in this QRS complex with the start of ventricular depolarization. Plateau phase of ventricle cells, and then all the way down here, this is our T wave, this is our ventricular repolarization. And of course, lots of things can go wrong with this, and the EKG will show cardiologists a ton of data. For example, what would a missing P wave imply? This is simple. This just tells you that your SA node is not working. If the P wave is missing, but you still have a QRS, it tells you the SA node is not working, but the AV node is working. That's an example of that ectopic focus. What happens if some of these segments are too long? What happens if the PQ segment is really long? That's going to imply your atrioventricular node is delaying the signal too long. There's an entire textbook written on how to read this, but that's just two examples. Problems with the conducting system often manifest as heart blocks, first, second, and third degree blocks, depending on how much of the conducting system is damaged. And this completes this section. Next, we'll tackle putting all of the events together: the EKG, the heart sounds, and the physical events happening during each cardiac cycle.